Welcome to our webinar, No More DMF, Greener Solid Phase Peptide Synthesis with Binary Solvents. My name is Mirko Lotz and I'm Group Leader Science Marketing at Bachem. It will be my pleasure to moderate our webinar today. I'm very happy to introduce Sandip, Jadhav, who works as R&D project chemist at Bachem. Sandip obtained his Ph.D. in peptide chemistry from the Indian Institute of Science, Education and Research, ISA Pune. And during his postdoctoral study, he gained experiences in the utility of peptides for tissue engineering and modulation of peptides. Sandip, moreover, is co-author and author of 2 scientific publications in the journal Green Chemistry in 2021, which are the scientific basis of the work he will present today. For the Q&A session, Caroline Lechner, Director R&D at Bachem, as well as Daniel Setcher Pedersen, Senior Development Scientist of our collaboration partner Novo Nordisk, will join us to answer your questions. However, before some housekeeping notes, your microphones will be muted during the webinar, but please write us in the Q&A session at any time your questions in the Q&A field in the bottom of the Zoom window. Before we continue with Sandip's presentation, we would like to share some information about Bachem for those webinar guests who do not know us yet. Bachem is a contract development and manufacturing organization specialized in the manufacturing of complex drug substances, namely peptides and oligonucleotides. We value our long-term partnerships with companies like Novo Nordisk, and we are proud of contributing by this to medicines which improve the life of patients globally. Bachem is committed to a long-term mission of leadership based on the continuous expansion of capacity for the production of peptide and oligonucleotide drug substances. By this, we serve our customers who use these products as key ingredients for medicines, which are used to treat a broad range of diseases, reaching from orphan applications to metabolic disorders, the latter with huge patient populations. A remarkable project in terms of capacity expansion we would like to highlight here is a new production plant for the manufacturing of peptide and oligonucleotide drug substances we are currently building. Our Building K will not only double our manufacturing capacity for peptide and oligonucleotide drug substances in short time frame, it will also lead to the creation of approximately 300 new jobs and will harbor innovative technologies which make the manufacturing of peptide and oligonucleotide drug substances more green and cost-effective. Bachem's business is built on 5 strategic foundations, reaching from people and culture to operational excellence and quality. Our webinar today will focus in particular on the aspects of innovation, technology, and sustainability, since Sandip will take you on the journey to showcase how we, together with our valued collaborators, Novo Nordisk, and the group of Professor Albaricio of the University of KwaZulu-Natal in South Africa, identified environmentally more benign solvents for the use in solid phase peptide synthesis. The latter is a common technology used for the production of peptide drug substances from small to large scale. And with these words, I would like to hand over to Sandip.
Thank you, Mirko, for the introduction. Hi everyone, I welcome all of you again to this webinar and also wanted to thank you for your time today. I will walk you through sets of very interesting experiments to convince you at the end, yes, it is possible to replace DMF in solid phase peptide synthesis. But let me explain very briefly why do we need to replace DMF in solid phase peptide synthesis. Peptides are very interesting biomolecules, and most striking feature of peptide is that they fold. They can adopt defined three-dimensional structure. And that's the reason there are more than 80 peptide-based active pharmaceutical ingredients, also abbreviated as APIs, in market. And there are more than 150 peptide APIs are being investigated at different stages of clinical trials. That means there is a huge demand of peptide supply every year. So how do we get access to the tons of peptide-based APIs every year? Thanks to the technology called as solid phase peptide synthesis and also abbreviated as SPPS. And this technology has been matured over the years, considering dimethylformamide, abbreviated as DMF, as a solvent which suits to all the steps involved in this technology. And now, why to replace DMF while it is already being used as a gold standard solvent? That is because it's no more secret that DMF is neither so friendly to human health and not to the environment. In 2005, ACS Green Chemistry Roundtable was established to promote green chemistry in pharmaceutical industries. In 2012, European Chemical Agency specifically categorized dimethylformamide as substance of very high concern. And it might be possible that more restrictions on usage of DMF is coming. Bachem is arguably one of the largest supplier of peptide-based APIs, and we always prepare ourselves to provide peptide APIs uninterruptedly. Therefore, we join our forces with our collaborative partner and very talented team from Novo Nordisk and known academic group of Professor Fernando Albercio. And this three-way collaboration is an excellent example of synergy between academia, biopharma, and CDMOs. And we set a goal in front of us to identify a solvent which can replace dimethylformamide in solid phase peptide synthesis. This might sound very trivial. However, the alternative solvent should qualify regulatory, commercial, and some technical criteria. Therefore, we laid a roadmap in front of us. Starting with solvent selection, we identified, together with Novo Nordisk, 19 green solvents, which are not restricted by European Chemical Agency. Then we subjected these solvents for further screening experiments, starting with solvent stability, followed by resin swelling. Then we also analyzed coupling kinetics and FMOC removal kinetics in these alternative solvents. And at the end, we realized two properties of solvents are pretty important. One is viscosity, and second is polarity. And we also identified at the end, when solvent matches this two property with DMF, could be best suitable replacement for DMF in solid phase peptide synthesis. And at the end, we demonstrated a DMF-free synthesis of bivalodin as a model peptide. Now, before going into much detail, let me explain which properties of solvent we are looking as a replacement for DMF in solid phase peptide synthesis. In general, the solvent should have reliable supply chain when we think about production scale synthesis of peptides. Again, solvent should not be very costly. It should be very easy to handle the solvents. That means it should show high boiling point and low melting point. And more importantly, it should not be restricted for their usage. More specifically, solid phase peptide synthesis involves several steps: resin swelling, solubility of reagents, coupling kinetics, F-MOC removal kinetics, and washing efficiency. And the solvent, which could best fit all these sub-steps involved in solid phase peptide synthesis, could be ideal replacement for DMF. And DMF strikes very well balance here and fits in all these steps involved in solid phase peptide synthesis. And therefore, we are looking for a solvent which can show similar polarity and similar viscosity as DMF. And we are also looking for solvent more specifically, which is polar aprotic solvent. As I said, several groups have already provided efforts to identify solvent which can replace DMF in solid phase peptide synthesis. But there is no niche solvent which can qualify or which can best support all the steps involved in peptide synthesis. N-butylpyrrolidone shows good resin swelling property, shows good solubility of the reagents involved in solid phase peptide synthesis. However, it shows high viscosity, which is not so suitable for industrial production of peptides. Coming back to THF and 2-methyl THF, again, it showed good swelling of resin. However, solubility of starting material and byproducts was not so ideal. Again, the F-MOC removal kinetics in these solvents was not ideal. Gamma-valerolactone showed better swelling. However, coupling kinetics and F-MOC removal kinetics was moderate, and solubility of reagents was a problem in gamma-valerolactone. Ethyl acetate showed good swelling. Again, the solubility of byproducts and reagents was not so optimal. Ethylene carbonate and propylene carbonate showed some promises. However, it was only recommended to be used for synthesis of short peptides. Then together with Novo Nordisk, we identified a set of solvents that can be used for solid phase peptide synthesis. For example, N-butylpyrrolidone, dimethyl isosorbide, gamma-valerolactone, tetraoxoandecane, styrene, dimethyl sulfoxide, 2-methyl tetrahydrofuran, tetrahydrofuran, 1,3-dioxylene, ethyl acetate, tetrahydropyran, ethylene, and propylene carbonate. And we subjected these solvents for further screening experiment. To start with, we wanted to know whether solvent is stable enough during peptide synthesis and does not get covalently reacted with growing peptide chain. To test this solvent stability, we incubated this pentapeptide in solvent at 40 degrees Celsius for 7 days and looked for any N-terminal modification due to solvent instability. To test how solvent behaves under acidic and basic condition, separate experiments were performed using HOBT and DIPA as additive in the solvent, respectively. Here are the results. DMF showed pretty good stability as a niche solvent and in presence of diisopropyl ethylamine, and here is the native peptide. However, when we incubated DMF together with HOBT under acidic condition, it showed the reaction with growing peptide chain, which is seen here as an additional peak, which is appeared due to the solvent instability. When we digged into more detail, we identified that DMF is not pretty stable under acidic condition, and it get degraded, and formyl group from DMF get transferred to the growing peptide chain, and which is not ideal for peptide synthesis. Similarly, we tested other solvents, and tetraoxoundecane and dimethyl isosorbide showed some reactivity towards the peptide. And we identified that peptides show deamination of N-terminus and methoxy group from the solvent getting transferred to the peptide chain, which is again not so ideal. Propylene carbonate and gamma valorolactone also showed instability, and ring of this solvent got opened due to the nucleophilic attack of growing peptide chain, as you can see here in the structures. Tetrahydropyran, tetrahydrofuran, and 2-methyl tetrahydrofuran showed pretty good stability under all tested conditions. NBP, which is N-butylpyrrolidone, also found to be stable. However, it was carried over into the crude peptide, and we speculate that might be due to its high viscosity. DMSO, styrene, and dioxazolane showed pretty good stability, and also ethyl acetate with minor percentage of N acetylation of peptide. With this, we move further with next screening experiments. Resin swelling. When we talk about solid phase peptide synthesis, or solid phase synthesis for that matter, one should know how the solvent swells the solid phase, that is resin. As far as solid phase peptide synthesis is concerned, it is important to know the swelling of not only starting resin, but also peptide resin. That is because with increasing peptide chain, the swelling propensity of resin can change, and that ultimately could impact the overall success of the peptide synthesis. We selected 4 peptide resins with varying chain length, starting with 8 mer octreotide, 20 mer bivalirudine, 29 mer glucagon, and 58 mer aprotinine, and tested its resin swelling propensity in selected solvent combinations. And also, for comparison, we tested the swelling property of their starting resin. In an experiment as shown on this slide, syringe reactor was installed on vacuum manifold, and solvent was introduced in the syringe where pre-weighed resin was already placed. After prescribed time period, solvent was drained from the syringe, as you can see on the lower panel here. What we have seen is that depending on solvent viscosity, some solvents took really longer to get drained off from the resin bed. And also, sometimes they cause resin beads to stick from inside of the reactor wall, and which is not so ideal for industrial scale production of the peptides. When solvent is more viscous, it might also take longer to get transferred from reservoir to the reactor through transfer tubings. And it could also delay the total reaction time, a total synthesis time. Then we move our attention to the next step to understand how good the solvent could diffuse through the resin beads. And we term this phenomenon as response characteristics. The resin swelling propensity of bivalirudine, which is 20 residue peptide, is shown on this slide. And corresponding resin swelling propensity of its starting resin is also shown for comparison. Green bar indicates the swelling of resin in given solvent, and bar in magenta indicates shrinking of resin to remove the solvent used for the swelling completely from the resin bed. The given resin was subjected for cycle of swelling and shrinking. For DMF, starting resin for the bivalirudine showed good swelling propensity, which was more than 4 milliliter per gram of resin. Bivalirudine, which is the peptide resin, showed moderate swelling propensity. We performed similar experiment for NBP, which is at least 4 times more viscous than DMF. NBP showed good resin swelling propensity for starting resin. However, for the peptide resin, it showed poor resin swelling propensity. Moreover, as you can see from this arrow, that NBP took longer to diffuse through the resin bits, and again, eventually it took longer to bring the resin to its highest resin swelling propensity, and which is again not so ideal for industrial scale synthesis of peptides. And here, DMF strikes very good balance of having optimal viscosity. Just to mention how peptide resin can swell differently than starting resin, the data of resin swelling propensity for octreotide, which is ATMA, is shown on the slide. With some exception, you can see peptide resin always showed less resin swelling propensity compared to its starting resin. Moreover, depending on the viscosity of solvent, we also seen a slow draining of the solvent from the resin bed and slow response characteristics. Also, few solvents, for example, TMSO and propylene carbonate, showed the phenomenon of sticking up resin bead on the inner wall of the reactor. And this is also not so ideal for the large-scale peptide synthesis. In solid phase peptide synthesis, we deal with two important reactions. One is coupling reaction, also can be termed as the amide bond formation reaction, and second is the FMOC removal reaction. Then we screen all the solvent to see how good they fit to support these two reactions in solid phase peptide synthesis. Starting with coupling kinetics, we coupled FMOC glycine on this tripeptide using DIC oxima coupling protocol. We isolated peptide after a prescribed time and compared the percentage of tetrapeptide versus tripeptide, which could give us the idea about coupling kinetics. And here are the results. As you can see, DMF showed moderate coupling kinetics. And in comparison to DMF, other green solvents showed either faster coupling kinetics or slower coupling kinetics. And we identified a trend. Depending on solvent polarity, coupling kinetic change. And what is the trend? With reducing polarity, we observed the faster coupling kinetics. And with increased polarity, the coupling kinetics got lethargic. Now let's see what happens with FMOC removal kinetics and how polarity can influence the FMOC removal kinetics. We subjected this FMOC protected tetrapeptide in 20% piperidine in given candidate solvent. And we analyze this adduct of piperidine with dibenzofulvene via spectrophotometrically. And you see the results here. There were only two solvents which showed the faster coupling kinetics. One is DMF and another is DMSO. And again, we identified the trend here with solvent polarity, albeit contrast with the coupling kinetics. Here, with increasing polarity, there was increase in FMOC removal kinetics, or FMOC removal kinetics was faster, which is again in contrast with the coupling kinetics. Now you can see how solvent can impart their influence on kinetics involved in solid phase peptide synthesis. From swelling experiment, we identified viscosity is a property of solvent, which is the important, and we are looking for the solvent with the optimal viscosity. And for reaction kinetics, we identified polarity also impart influence on the kinetics. And here, DMF strikes really good balance. Is there any other solvent which has similar polarity and similar viscosity as DMF? Let's take a look. Let's talk about viscosity. In comparison to DMF here, other neat green solvents showed either higher viscosity, shown in magenta here, or lower viscosity, shown in blue color here. We could not identify a solvent which showed similar viscosity as DMF. How about mixing two solvents together? One solvent with high viscosity with solvent with low viscosity. Whether it is possible to modulate the viscosity near to DMF. That's what exactly we did. And indeed, it is possible if we mix two solvents together in an appropriate ratio, it is indeed possible to modulate its viscosity very close to DMF. Now let's see how we can modulate polarity of solvent. We required certain polarity threshold for reagent solubility, solubility of isopropylurea byproduct, and also we need polarity threshold for the good epochremal kinetics. And again, DMF strikes very good balance here. In comparison to DMF, for example, here dioxazine shows lower polarity and DMSO shows higher polarity. When we started mixing DMSO in dioxazine, stepwise, we realized that yes, it is possible to modulate polarity of binary mixture very close to DMF. And this is how we can show that solvent characteristics can be changed to very close to DMF by mixing them together merely. We relooked into coupling kinetics in binary mixture and realized that yes, binary mixtures are better compared to neat solvents. As you can see, this area is more populated here, and solvents highlighted in green boxes shows faster coupling kinetics. Similarly, for epochremal kinetics, we have more possibilities of solvent which can give the faster coupling kinetics. Earlier with neat solvent, there were only two solvents which showed faster faster epochremal kinetics, one DMF, another DMSO. But now due to the modulated polarity, we have more possibility of solvent combinations. Our learnings from all this screening experiment are condensed in this polarity viscosity plot. We also call this as a PV plot. We hypothesized that if one wants to replace DMF in solid phase peptide synthesis with other solvent, the other solvent should show similar polarity and viscosity properties as DMF. However, we could not identify many solvents as a neat solvent which could show similar PV properties as DMF. However, as we have seen earlier, when we mix them together in a binary solvent, there are a number of possibilities of solvent combination which could show similar PV properties as DMF. Therefore, we hypothesized that binary solvents might be more viable than any neat green solvent to replace DMF in solid phase peptide synthesis at room temperature. Again, we hypothesized a solvent combination which can occupy space near to DMF on this polarity viscosity plot would be more beneficial compared to the solvent which occupying space away from DMF on this polarity viscosity plot. Therefore, we decided to synthesize B-valerdine as a model peptide in a solvent combination, DMSO dioxazine 327 and DMSO 2-methyl-THF 327, which is placed near to DMF on polarity viscosity plot. And we compared this result with the synthesis using solvent NFM dioxazine 228 and NBP dioxazine 228, which are placed away from DMF on this polarity viscosity plot. This is how we synthesized B-valerdine as a model peptide in alternative solvent. We started with leucine preloaded chlorotityl resin and subjected this resin for several SPPS cycles. Epmoc cleavage, washing, coupling, which was again followed by capping and again washing. The advantage of introducing capping after coupling step is that one can identify the difficult coupling and one can identify the difficult Epmoc removal stages. We synthesize bivalidine in solvent combinations alternative to DMF and compare the results with DMF. And here are the results. As you can see, for DMF, which could provide crude purity of bivalidine of 73.8% in comparison with this solvent combination, DMSO2-methyl-THF327 and DMSO-dioxazine-327, which were occupying space near to DMF on polarity viscosity plot, showed comparable purity of bivalidine. However, as you can see here, N-methylformamide NFM-dioxazine-227, that is N-formyl morpholine dioxazine-228, sorry, and NBP-dioxazine-426 showed compromised crude purity. And remember, they were placed away from DMF on polarity viscosity plot. With this slide, we could show the utility of our PV plot for preliminary selection of binary solvent. Again, there is always chance to improve the solid phase peptide synthesis by process optimization. We realize certain couplings need to be improved, for example, arginine coupling, and also, as you can see here, the impurities which are related to lethargic Epmoc removal also need to be improved. With this, let me summarize today's webinar. In collaboration with Novo Nordisk, we identified 19 green solvents as a possible alternative to DMF in solid phase peptide synthesis. Then we showed that solvent stability is also important when we are looking for alternative solvent as a replacement for DMF. And if solvent is interacting itself to growing peptide chain, it would not be ideal case. We learn about resin swelling for both starting resin and peptide resin. And we realize that viscosity plays an important role here, and we are looking for solvent then which shows the comparable viscosity to DMF. We looked more into coupling kinetics, where low polarity of solvent showed better coupling kinetics. On contrary, high polarity of solvent showed better Epmoc removal kinetics. Then we condense our learnings into simple, easy to use polarity viscosity plot. And we showed that merely looking at plot, we can pick the solvent combination, and you can speculate which solvent combination could be better replacement for DMF in solid phase peptide synthesis. Again, further process development might require. And this we have shown by synthesizing bivalidine as a model peptide, where we completely avoided use of DMF in all involved sub-steps of solid phase peptide synthesis. Is bivalidine only peptide that we synthesize in alternative solvent? No, there were more peptides synthesized in collaboration with Novo Nordisk, namely Junge-Rademann, EIB-enkephalin, thymosin alpha-1, and dasiglucagon. If you would like to know more about this synthesis and more about resin swelling properties of resins which are different than polystyrene resin, and also solubility of different reagents and coupling reagents in alternative solvent, I would encourage to go through this open access publication, which we published together with our valuable collaborative partner, Novo Nordisk. With this, I thank you all for your kind attention, and I'm looking forward to answer your questions together with my colleague, Dr. Caroline Lechner, and our collaborative partner, Dr. Daniel Sejar Pedersen. Now I hand over this presentation to Mirko, where you will have possibility to take actively part by answering poll question. Thank you very much.
Thank you very much, Sandip, first of all, for presentation of this impressive results. Just a remark for our webinar guests. So the presentation so far was pre-recorded to secure an optimum presentation quality. But now we go to our live Q&A session. Before we answer your question, we would start with a question to our audience. So we would like to have your opinion on what are your main concerns regarding replacing DMF. So do you have concerns about the stability of the solvents? Do you wonder if the resin swelling or the reaction kinetics would be comparable to DMF? Do you think side reactions could occur in the stages of the SPPS, or are you concerned about reagent stability? Or do you think that large-scale SPPS and binary solvents is still a difficult topic, or that costs are a topic? So please give us your feedback by using the poll tool. Waiting now for the results. So the poll is still ongoing. Okay, so now we have it. So it's very interesting. So almost half of our webinar guests consider that side reactions could occur during the stages of the SPPS. Or the second most important topic for our guests are reaction kinetics, if these are comparable to DMF, and also the solubility. So this is really very interesting feedback. Thank you very much. And maybe we can address this even more in our Q&A session. So then let's have a look what we receive as questions. Let's see. So one of the questions for Sandip, I think that's for you, is why were only polystyrene-based tested in the study? Or were there only polystyrene resins tested in the study?
Exactly. We tested not only polystyrene resin, but other resins also, and you will get more information on the other tested resin in the publication. However, we mainly focused on the polystyrene resin because they are more relevant for the industrial scale synthesis of peptide. They are more robust and they have sustainable supply, and cost is not high.
Mm-hmm. Thank you very much.
Answer.
Yeah, so it's mainly for economic reasons, for example, why you focus on them. Okay. There's another question. How you see in general SPPS as the future for peptide synthesis, or how you in general see also the application in green solvents in SPPS. I don't know, Caroline, do you want to take this question?
Yeah, sure. Thanks for this question. Yeah, I think there's also for SPPS and also like alternative solvents and SPPS, there's a great future for this one. And I think the question also comes from like there's a background. If you would compare SPPS to LPPS, there are already different technologies that are kind of had already the change from the kind of restricted solvents to a more environmentally friendly ones. However, I would say you should not kind of compare like solution-phased approaches towards peptide synthesis with SPPS, because they target completely different peptide targets. But I think both approaches have a great future.
Thank you very much. There's one question for Novo Nordisk. So Daniel, I think this is very interesting question also for you. So how is it with Novo Nordisk? So do you see that you will switch more and more to green SPPS processes, even though maybe the costs might not be so advantageous? How do you see this from your perspective?
Yeah, okay, so that's a great question, and I'm actually quite happy I got it, because we are very actively doing something about this. We have initiated an internal project, quite big one now, with a lot of focus on making actually, it's very ambitious in the next couple of years for new projects, I should say, that come to CMC. The ambition is to switch 100% to green binary mixtures. So we are actually in the process now of figuring out how to phase. We have a transition period, right? Where we use both DMF and green things. So we're in the process now of figuring out how to do this in a smart way. We have to retrofit some equipment. We have to, there's all these logistical issues, but it's happening. We have the manpower for it. We have the funding. So it's great to see something like this translate into a real life outcome. So this is actually a real thing and not just an academic exercise.
Thank you very much.
And then you asked about cost. That's a tricky question. We can accept a cost increase, but the question is how much? And it's not easy to answer. I mean, the systems we're looking at now looks good in terms of cost. We're willing to, some of them are a bit pricey, but we believe we can get the price down with increased demand. So we're quite confident it will work for what we've identified so far. There are also some supply chain considerations we're evaluating now. It looks good. So I think this is happening. It's not an open question anymore. We are doing it.
Yeah, very, very interesting to see. And as you say, very, very nice to see that these studies really translate into big applications and to see the aim also at Novo Nordisk behind. Very nice. Sandip, here's one question regarding side reactions. So how do you see this in alternative solvents? For example, racemizations of amino acid derivatives an issue, as our guest asked?
Yeah, first of all, thanks for asking this question. This is really interesting, and I'm really happy that our audience is really curious about side reactions. Yes, indeed, we looked into it. And if we talk about side reactions, there are also chemical reactions. And then, as we have seen, the impact of solvent polarity on coupling kinetics and epoxy kinetics, we also evaluated impact of solvent polarity on side reactions. And more interestingly, binary solvent, we can modulate the polarity, and they prove more beneficial compared to DMF to contain the side reaction, let it be racemization, amino acid racemization, or aspartamide formation. And indeed, we have also looked into the arginine lactamization, and we could also show that just modulating the solvent polarity by mixing them together merely, we can contain side reactions. And I think this could be a less invasive way to contain the side reactions just by changing the polarity of solvents.
Thank you. Here's also a very interesting question. So how is it about the recovery of binary solvents? Are there any studies available? Caroline, do you want to take this question?
Yeah, sure. Yeah, it's an interesting question, I think, and this is also a kind of a topic that is in the field of all this like greening chemical processes, for us also greening SPPS processes, another aspect. So what we presented today and what was our focus and really finding an alternative solvent for DMF, but of course we also have a look, maybe not that much progressed so far as this study yet, but we also have a look on the different other aspects. For example, like would it be able to recycle these solvents? And this, of course, then also depends a lot on the physical properties of your solvent or solvent mixtures, because we recommend binary solvent mixtures, and the difference, for example, in boiling points. So, and then it would, in principle, be possible to also recover and eventually also recycle these solvents. However, I also have to say, as like an CDMO, and we also manufacture a lot of a GMP, like reusing recycled solvents for the GMP is also has the compliance aspect one has to also consider. But could be reused for, of course, for other purposes or as a research grade solvent or solvent mixture then, and also to lower kind of the environmental footprint.
Mm-hmm. Okay. Thank you very much. Let me see what. There's one question. I think Sandip fits good to you. How far were the parameters of the post-D protection wash steps optimized in the study? Can you comment on this? So, like number of washes, wash times, and
specific for the. Can you repeat the question?
Where the parameters of the post-D protection wash steps optimized. So I think it's the washing after D protection. Yep. If you optimize this regarding the number of washes and the composition of washing solvents, so was this part of the study?
Yes, it is also mentioned in our publication. However, I will comment very briefly on it. We optimized washing steps until we have the minimum amount of piperidine found in the wash solvents. So actually, we did not to put a lot of effort on it using our typical washing regime. We observed that we could really bring down the residual piperidine or residual base that we use for the FMOC removal to the really minimal level, and which was, again, we found that even better than DMF. And that's how I think binary solvents, as we say, we unlock the property, or we unlock the potential of binary solvent. We really optimize every step just by changing the combination of solvent mixture. And we can make sure at the end that every step in the SPPS was optimal.
Okay. Thank you very much. Also, one more question. It's also regarding the chemical experiments, or let's say the use of the study for all sequences. So we have the question: what happens when we have methionine in the peptide sequence? Did you try this? Is there some specific things you would note in this regard?
Yeah, exactly. This was also our concern in the beginning, whether this kind of sensitive residues will sustain the oxidation. But what you have seen is really interesting. Methionine was never found to be oxidized, even if we use DMSO as a component of the binary mixture. And then we also gave a thought, even if DMSO oxidizes methionine, there is a possibility that we can reduce it again in the cleavage, optimizing the cleavage condition or cleavage cocktails.
Okay, thank you. There's one question, I think, Daniel, that goes out for you. So when you say Novo Nordisk is implementing this green solid phase peptide synthesis processes, and so far these studies concern more the toxicity. Is there something with the CO2 footprint in terms of sustainability you have to consider with the solvents as well?
Yes. So this is, of course, that's a good question. Let's how to answer it best. I mean, it's SPPS. What we've done here does not really address the CO2 footprint. The main objective here was to find a short-term solution to eliminate toxic substances, toxic solvents that constitute the vast majority of raw materials used for SPPS. So we've done that now. We are actually now at Novo, and we are talking with Bachem, have early talks about this now also, trying to see if we can lower the CO2 impact of SPPS. That said, it will always be high, but we are trying to cut down on it. Specifically, we are trying to implement new washing procedures where we alter the ratio after, in particular, FMOC removal, which requires less washing, and to put in some inline monitoring using some PET technology that will tell us exactly when pyridine levels are at the level we want them so we can stop washing. And I think a very conservative estimate is that we can at least cut 20% off the solvent consumption using these approaches. But I mean, this is work in progress, and we also, we have to retrofit some equipment. We have to discuss with the CMOs we collaborate with, can they implement these things and so on. But we hope we can do something here. If you really want to do something about this, then someone has to reinvent peptide synthesis. And this is a very, it's not something that Novo Nordisk can solve. This is a very ambitious, very long-term project. 10 years down the line with lots of funding, maybe. But for now, we do what we can for now.
Yeah, very interesting topic. Thank you for these insights. We have one more technical question. So how would the coupling kinetics be affected by heating the reaction? Is this something you tested or investigated as well? Sandip?
Yeah, I think I can better answer this question. Yes, I mean, as for not only for coupling kinetics, for any chemical reaction, if there is always a parameter of temperature which influences, and with increasing temperature, there is increasing chemical kinetics, or for example here coupling kinetics. Yes, we observed in separate experiments. Yes, with temperature there is a certain boost for coupling kinetics. However, for this project in particular, we were aiming to find a viable solvent that can be used at room temperature, because we wanted to do this for industrial scale production of peptide, and not only for the academic purpose. So with increasing temperature, I think there are also some kind of side reaction which will be for sure aggravated. And when we talk about kilogram scale of peptides, resins are being synthesized, and this side reaction contributes to the impurities, it won't be an ideal case for large scale synthesis. We still prefer large scale synthesis of peptide at room temperature.
Yeah, maybe I can also add here. So like, this is absolutely true. So of course we are aware of like the that you can have a benefit of running several reactions, or in the whole SPPS thing, some reactions at elevated temperatures, as Sandip mentioned. So you can also have a lot of drawbacks, and then you have to go for your peptide target and really develop and have a look individually on this. Do you have a real benefit, or do you also have like a drawback that would also eventually affect your product quality in the end? But of course you don't want to have. So, but we always have these, or we try to have like all the different aspects in the focus, and we then choose the best compromise between all this. And also what to add, for example, but we did not test this. Of course, we also, what Sandip mentioned, so like solvents with higher viscosity, or neat solvents with higher viscosity, were kind of then also excluded from our studies, but we are also well aware that you can tune or lower viscosity by kind of giving some heat to the reactions, for example. Also, but again, this is also then very specific and has to be kind of investigated and balanced to find the best solution.
So the approach here was really the investigation for room temperature first.
Exactly. Really, that we can go, because we really not, of course, we also do like feasibilities and small scale studies usually if we start with a new project together with our customers, but then we also aim in going for like large scale SPPS, and to serve, provide like a peptide APIs.
Exactly. Just quickly quickly adding to Caroline's answer, yes, and also it would also be much depending on the peptide, which kind of peptide you want to synthesize. Is peptide sustain the high temperature? I think it would be a good idea. It's really depending on the peptide to peptide. If there is a less chance that peptide could give any considerable side reaction, then yes, it is still a good idea to have higher temperature for coupling and even for a macromole.
Okay, thank you.
Maybe I could also add a comment here. Because I didn't mention it before in terms of CO2, but we actually also see some relatively easy to evaluate if it could also improve washing procedures. So actually have your washing solvents preheated. And then by using heated solvents, you might actually also, I mean, logically, you would think you would be able to reduce also just by heating to maybe 40 degrees or something. And that's quite non-invasive way of heating, right? Because it shouldn't lead to side reactions. You wouldn't think so.
Thank you. There's one more question. It addresses the IP situation of the studies. Are there any patents or is there some IP restrictions, or are there patents under filing? So maybe Caroline, this is something you could answer.
Or together also with Daniel, I think so. Thank you. So no, we published. So all what's published, it's not patented and not IP protected.
That so was really the plan from the beginning. I mean,
exactly.
We would really like to see this get widely adopted. Again, something with supply here. I mean, if everyone suddenly wants to buy some of these alternative solvents, the community will benefit by cost coming down quite fast. So,
exactly. It was really not our aim from the beginning, really to have like any IP. We really wanted to do the research and to share it also with the community to get it done.
Yeah, and it's a good remark also for the cost situation, what was already addressed. So when it gets more common, the green solvent usage, that you also bring costs down at the end. One more technical question. So DMSO is used to form disulfide bonds in peptides. So how can one reduce the formation of dimers or cyclization using DMSO in binary solvent mixture? So is this something you investigated and had a look at, Sandip?
Yes, in typical DMSO is used as an oxidizing agent for formation of disulfide bond. And that was also our concern from beginning, whether it might impact the growing peptide chain. However, as you can see in the synthesis, all side chains are protected and they're less labile for the side reactions as far as cysteine side chain is concerned. So when side chain is protected and throughout the SPPS and there is DMSO around, we did not see any disulfide bond formation or any oligomerization for that matter.
Okay.
I could maybe also add that we evaluated this. We evaluated some binary solvent mixture where one component was DMSO on a Novo Nordisk phase 3 project actually, and that has a disulfide bridge, and that we didn't observe anything, no problem at all. So this is not a concern, at least not on these targets.
Perfect. So I see we're getting to the end already of our Q&A session. Maybe one last quick one. Were all reagents soluble optimal in the binary mixture? So did you see any from the process, any drawbacks with the reagents you use in the solvents, solubility wise?
Yes, I think I could answer this question. The first thing first, we looked into the solubility, which is really important even before you go for the SPPS. We need to know whether your reagents are soluble. We obviously found some difficulties in solubilizing certain amino acid with certain side chain protection or even N protection. Then we have a toolbox of solvent. If some solvent combination is really not good for certain amino acid, there is another combination which is coming into the rescue. And then we have developed a solvent toolbox. But in general, we did not face really a big problem when solvent, the reagent concentration is around 0.1 molar.
Yeah. Thank you very much. Thanks a lot for all the questions and also the answers. So I see we are at the end of our webinar already. So yeah, first of all, once more, Sandip for the presentation. Thank you very much. Caroline and Daniel also for being here in our Q&A session. And of course, thank you very much to all our guests in the webinar for joining us. One remark, if you want to learn more, please visit our website. So here you can find the publications we have together with Novo Nordisk on green chemistry. Please also go to our blog. So there's also a blog about our collaboration with Novo Nordisk and these studies. And do not miss out to look also in our brochure showing what services and products we offer for our customers. And yeah, thanks very much once more for joining us, and very happy to see you in our next webinar. Thank you very much, and bye-bye.
Thank you.
Thank you.
Thank you for the questions. Bye.
Bye-bye.
Thank you.